So it's a pleasure to introduce our first speaker this afternoon, Manuel Salto Tellez from Belfast. Manuel has got a very interesting curriculum vitae. He trained in medicine in Spain and Germany and Leiden in the Netherlands. He went on to train in histopathology or surgical pathology in the UK, and then a molecular pathology fellowship in the USA. He then went from that into a very distinguished role in Singapore, heading a very large molecular pathology laboratory and project, and then lucky Belfast managed to poach him to become their chair of the molecular pathology project and also a consultant pathologist in the city. Manuel is a true clinician and a scientist who combines these two activities together, and he's going to tell us today about digital pathology in biomarker validation and diagnostics delivery. Manuel.
Thank you very much, Darren, for a very, very kind introduction. I was saying earlier on that perhaps at a time in which digital pathology is fashionable, we should pay credit to those who held the fort in the days in which it wasn't so fashionable, and our the two gentlemen at the table here are a credit to that. These are my disclosures. I would like, first of all, to really thank the organizers, David and Nazir, for the kind invitation, and also congratulate them for what I think is a wonderful program. You will hear later on in this program that there's been significant investment in the UK in digital pathology. The Path Lake is one of those examples, and we are really looking forward to working with David, with Nazir, with the rest of the academic partners, and with Philips in what potentially could be a program that could transform the way we do pathology in many ways. It's going to take you 10 seconds to realize that I'm not a computational scientist, and perhaps another extra 5 seconds to realize that I'm not a computational pathologist. What on earth am I doing here then, you may wonder? Well, for years we've been trying to put together a program that would try to bring together everything that we think is necessary today in pathology. Anything from biobanking, traditional pathology, bioinformatics, digital pathology, molecular work. And this is truly integrated for several reasons, because it integrates, as you can see, technology that usually you see in different laboratories, because you are integrating hospital and academia. We have a laboratory that is accredited to do diagnostics, but is heavily involved in translational research, and we are essentially in the process also mapping what we think is the way in which these things should come together into a single program. So as you can see, perhaps in a contrast to what you're going to hear today, my presentation is going to be very, very practical. I hope not too much. We want to bring these things together because we acknowledge that this is the way pathology has moved in the last few decades. Since pathology became a clinical discipline, we would like to argue that there's been 3 revolutions in pathology. The first one is when pathologists adopted immunohistochemistry. This was extraordinary, the fact that pathologists were able to look at intensity of biomarkers, subcellular localization, tissue specificity. In fact, in some areas, we invented a totally new taxonomy of diseases based on this. And we are acknowledging now that apart from that descriptive pathology, there is also very much a molecular immunohistochemistry, very much therapeutic, which probably should be delivered with different rules. The second revolution has been the revolution that started or should have started with the discovery of the structure of the DNA, but has come to us significantly later, mostly with the development of next generation sequencing. And as you know, the activity of pathology departments in this area has been varied, to say the least. We think that there is a third revolution going on, and when in 10 years or so we look back, we probably remember these years at the time in which pathology, digital pathology, artificial intelligence became a reasonable identity for the way we practice pathology. The promise and obviously the expectation right now of what artificial intelligence can bring to medicine is phenomenal. I don't know how much of this really is workable or not, but we are saying that we can apply artificial intelligence to the analysis of complex genomics information, to the analysis of complex clinical imaging like radiology or indeed pathology. We are beginning to say that artificial intelligence can go into the clinical notes of our patients and make therapeutic decisions that perhaps are better informed than the way clinicians are doing it today. We know that we can complement that with the history, with the web history of individual patients to understand social trends and to start bringing some other work together. And of course, we can take that individual patient, take it into a population-based information, and start deciding again on potential therapeutic avenues. All this is the promise that we want to translate in prevention, in understanding predisposition, in diagnosis, in therapeutic intervention, in the effect of the individual into global health, you name it. It's very clear that of all these options, perhaps tissue pathology is one of the ones that is more advanced. From an extremely practical point of view, and if there is a message perhaps I would like to give you today, is that remember the artificial intelligence activity and the tool that comes from it is measuring what at the end of the day is a biomarker. And the rules to identify and to bring those biomarkers into diagnostic or clinical practice are actually very specific. You need to understand that we are working with a biomarker, that this could be a bona fide companion diagnostic, and that may change what we understand is today the ground truth that we are bringing to develop and to deliver these biomarkers. This is the way we explain in the Oxford textbook of oncology, the way we explain the oncologists what is a biomarker. Is anything that can be measured and that is going to help you diagnose or decide on therapeutic intervention. As you can see, most of those biomarkers are actually applicable to tissues. We should be optimistic. And yet, we have to be very careful. Why? Because until today, the work that we have done in biomarkers have been very lousy. Of more than 100 studies out there that claim that there is a clear diagnostic or a clear therapeutic opportunity, less than 1% make it into something that is actionable, that is druggable, that is usable today in our patients. And the reason perhaps is what we have called the filters of reality in biomarkers. If you take into account those biomarkers that we really don't understand, those that were not developed with robust technology or that were developed in designs of studies that were not adequate, or very important, those that were validated suboptimally, probably that explains the huge attrition rate that we have today. This needs to change. And it needs to change like we argued with Peter Hamilton and Jackie James in a paper 2 or 3 years ago in the context of clinical trials. That is the measure of the value of a biomarker. And that is where should we start bringing our artificial intelligence tools to make them companion diagnostics in the way we are practicing medicine. And just the last a consideration in this general review, and this is again a reflection that will be putting together very soon with Ian Cree. We are actually the taxonomies of disease. Much of the ground truth of what we do comes from the pathology analysis. And yet, it is very likely that what is going to happen when we start bringing artificial intelligence into this analysis is that we are going to have to revisit that ground truth. Because remember, the goal is not to be as good as the pathologists are. It's to be better. Because of that, and if you start putting together what it is the most simplest way in which pathologies operate, remember, we always start with an H&E. The vast majority of the times that leads into a diagnosis. More and more often we use tissue hybridization-based techniques, immunohistochemistry, in situ hybridization. Some of these are interpretative, but more and more often, as I mentioned earlier on, we are getting into quantitative decision-making. And more often as well, we are annotating samples for nucleic acid extraction to move into molecular diagnostics. I would like to argue that digital pathology today is potentially embedded in all these processes and more. We showed a couple of years ago that you can use digital pathology to better identify those areas ahead of molecular diagnostics. You will see several examples today of how artificial intelligence digital pathology can quantitate biomarkers in a way that is superior to the average pathologist. We are beginning to see, and the next speaker is a world expert in this area, how we can begin to extract information from H&Es that are substantially better than the way the naked eye pathologists can do it. And surprisingly, we can begin to have predictive molecular digital pathology. We can begin to understand what is not only the molecular status of a disease, but also the topography of where those mutations are in a specific tissue. Only this is opening avenues that have been unthinkable until now. Some of these avenues are avenues that we have pursued in my laboratory, and what I'm going to show you is some of that work. First of all, in some of the technology adoptions that we've had in this area, and then on how this has been applied to some of the clinical questions in cancer. So it's going to be 3 technologies very briefly and 2 clinical questions. If you're interested in others, by the way, remember there are other 3 posters from our group telling you much more about digital pathology of PD-L1 that I'm going to show you today, telling you about a very interesting relation between proteomics and transcriptomics in ovarian cancer, and showing a first example of how digital pathology can be predictive of malignant transformation in oral dysplasia. Very basic. What is the elephant in the room if you want to apply next-generation sequencing in diagnostics? It's very clear. If you want to do broad next-generation sequencing, you are going to have a significant attrition rate. And we have many examples in the UK showing that that is the case. The quality, but also the quantity of the DNA that you put in your system is essential. In fact, in my laboratory, we don't do any form of high-throughput analysis unless we understand what is the nature of that sample. And like our colleague David Gonzalez has shown, there is a very direct relation between the amount of cells that you put in that analysis and the failure rates that you get in next-generation sequencing. The problem is that pathologists have been notoriously bad at telling any molecular diagnostician what is the content of that tissue. Not because we don't know how, probably because we never agreed what it is that we are actually scoring. So very simply, years ago, together with Peter Hamilton, we developed a very simple digital pathology approach that is telling us where the tumor is and how much tumor there is in that area. If you take that simple approach to what industry is doing today in this space, it becomes phenomenal. So this is one of those cases that are very fashionable for us today. High-grade colorectal cancer, lymph node with significant lymphocytes, stage 4, PD-L1 positive, amenable for immune checkpoint inhibition. And this is how digital pathology, artificial intelligence tell us today what are the different components of that tumor ahead of analysis. Is quite frightening. QPath was a system that was developed in our laboratories by a very bright mind that you're going to see in this meeting, Pete Bankett. He developed it in Queens, and now he's developing it further in a very distinguished academic career in the University of Edinburgh. Pete developed a phenomenal tool, not only because he's very good at what he does, but it also took into account from the beginning how the user approaches the analysis of a tissue in silico. QPath is very good, has a whole slide view which is second to none. It's very good at scoring subcellular signals, has a system to score tissue microarrays and a system to recognize tissue up front. It works with many different images. It gives you real-time understanding of what is the clinical relevance of what you are doing. It's not surprising that up to March of this year it had 28 downloads. It's highly cited, and as you can see, not only in high impactful research, but also in comparative analysis of different tools, QPath, it becomes very, very highly rated. When we had this tool, the first thing that we wanted was actually to show that it did what it said it did. In this particular case, with more than 700 colorectal cancers, we were able to show the survival advantage of CD3, CD8, a tumor suppressor gene, and an immune checkpoint inhibitor, the relation in survival between the percentage of stroma that you have. In 300 breast cancers, we were able to show the advantage of the scoring in the relevant breast cancer biomarkers, including Ki67, that, as you know, is notoriously difficult in this area of digital pathology. We've approached multiplexing probably from a much more less sophisticated way than others have done. We went to very specific questions. We wanted to confirm that the signal that we identified in our research was in very specific cell types, to confirm and to improve on where we feel are the shortcomings of traditional immunohistochemistry, and to refine the interpretation of digital pathology. I'll give you some examples. So this is from an esophageal cancer study. We had evidence that ICOS is a very important prognostic factor. We wanted to see clearly that the signal came from the immune cells and not from the epithelial cells. Here you have another study. We were interested in TGF-beta1. As you know, cytokines, extracellular proteins are not very good at immunohistochemistry format. Here you can see a mixture of RNA-ish and protein in the same analysis. And here you see something fresh from the kitchen of Matt Humphreys, PDL1. Those of you that are involved in that scoring, you know how difficult can it be. Where is the signal coming from? Many times you wish you have dual staining to understand if it came from epithelium or from immune cells, and indeed this is what multiplexing can do. How do we apply these then to very specific questions? The first thing that we did was to apply to a large study in PDL1 in lung cancer. What Matt and Stephanie could show here, in an area that, as you know, is very complex. PDL1 is probably only one element of several that are trying to tell us how to start using immune checkpoint inhibitors, and probably the expression of PDL1 together what we call today tumor mutation burden, or the innate immunity of the patient are probably equally important. In any case, the problem of PDL1 is it's a very lousy test. Among other reasons, those of you that have used it, you will know that it is among other reasons because unlike other tests, for instance HER2, where you have a buffer zone of 2+, and you can use a genomic analysis of HER2 to tell you where the patient lies, PDL1 has very narrow therapeutic thresholds. And you reach to areas that are really pretty difficult. For instance, imagine that you have a case where the ground truth is 53%. If you think as a pathologist that the score is 97%, you're probably right, because you are in the right therapeutic threshold. But if you think it's 47, you're wrong, because you are in the wrong therapeutic threshold. This is the problem with tests that are not very well defined. And again, what Matt and Stephanie could show in this paper is first of all that the quantitation of RNA scope in adenocarcinoma had a very good concordance, as you can see, with the results that we got in diagnostics, but also that if you apply QPath, you begin to have significant degree of concordance, except, of course, when you are very close to your thresholds, and then the question is, what do those thresholds really mean? Another important contribution, I believe, to this area of biomarker analysis is the work that Stephanie, Matt Allardyce, and Matt Humphries have done as well in this area. So Stephanie was interested in going back, looking at 12 biomarkers that we know are important in adaptive immunity as immune checkpoints, and try to understand what was the prognostication value of those markers in colorectal cancer. And for that, she applied 3 very well-defined clinical cohorts, a cohort of stage cancer 2 and 3 from Belfast, a cohort of stages 1 to 3 from Aberdeen, and a clinical trial cohort stage 4 coming from a group from the UK, the FOCUS trial. In total, more than 600 cases. And what she showed is that adding CD4 to the mix of CD3 and CD8 significantly improve the prognostic value of this analysis in stages 1 to 4, including stage 4 colorectal cancer. It probably has predictive value as well, but most importantly, she identified the reason why this is the case. In other words, the immune silent group is actually defined by a very specific transcriptomic signature, by a very relevant hypoxia signature, and by very specific association with biomarkers like SPP1. In other words, we know why digital pathology is being superior in telling us what is the prognostic value in different cancers. So I hope I've been able to show you that indeed the promise of artificial intelligence is very relevant, is very important in general, but I think there is a lot of promise in the work that we do in tissues. There is another promise, and it's the promise of what we are doing in research. Because remember, for years, for probably more than a decade, we were trying to understand how we build system biologies in our cases. Is it genomics? Is it epigenetics? Is it transcriptomics? Is it proteomics? Is it metabolomics? Is it a mixture of those? What takes precedence? What is hierarchically important? How do we start looking at this complexity? Wouldn't it be extraordinary if at the end of the day is actually the tumor ecology and the way the immune contexture and the contexture of cancer is associated morphologically, what really makes sense of all the molecular information that we have? We are beginning to think that that is the case. And that would also be a great contribution, not only to the way we are applying biomarkers clinically, but also to the way we understand disease. Allow me to finish then acknowledging everyone that has been involved in this work, including and highlighting particularly Jackie James, who has been co-leading this program with me for years and to whom I'm indebted, to those that externally have been helping in our research, and of course, those who pay our bills at the end of the day. Thank you very much.
Thank you, Manuel. Fantastic talk. My assistant is getting ready with the microphone, so get your questions lined up. Anyone in the closest bit to me that wants to ask a question before we get that set up? Jason has a question.
Manuel, it's beautiful and amazing work.
Just remind us who you are for the rest of the room.
I'm sorry, I'm Jason Swiller from the University of Dundee. Most of your talk talks about using specific biomarkers, and those biomarkers are based on biomedical knowledge, et cetera. In your last slide, you're opening much, much farther up to not only many modalities, but you're implying a much broader sampling of biological space. And so there's a very different way of thinking about how to work with these signals. Am I reading that right? Is that the anticipation?
Yes. So we are always saying that if your ground truth is the pathology opinion, you'll always be as good as the pathology opinion, never better. If your ground truth is how existing biomarkers operate, you're going to be as good as what that biomarker allows you to do. I think we need to open up. I think we need to start integrating information that individually we not even know what it means. That will be the great value of artificial intelligence, in my opinion.
Thank you. Another question? Time for one more, I think. Anyone at the back want to ask a question? I might just ask one for myself, which is you touched on multiplexing. So a lot of us think that the potential of multiplexing and digital pathology are a very nice marriage. What are your opinions on that, and when do you see it happening?
I think so. So our early evidence tells us that it's probably more difficult to validate than we originally thought. If you take as ground truth the scorings that we get with uniplexing, it takes a while to make sure that works. What I think that multiplexing has is a potential that we haven't seen in other areas from a very practical point of view, and is the therapeutic value of co-expression. We haven't tested that in our biomarkers until now, and I think that is potentially a very important area, which I'm sure is going to have therapeutic implications.
Well, thank you. And thank you again from all of us.
Thank you.